Well, we were looking at dinosaur fossils. The bones have been fossilized from dinosaurs. And these honestly look more like rocks to me, but they're, what do you have here? What are you studying?
Well, this actually is not a bone. These are fragments of a Triceratops horn.
Okay.
In 2012, the Creation Research Society sponsored Mark Armitage and I to go to the Hell Creek Formation in Montana, which is a very popular place for finding dinosaur bones, and we instead dug out a Triceratops brow horn. Now, it's just in crumbled pieces now, so we can't really, put it together and show you a horn. And what we've done, of course, is have to work with it where you actually destroy portions of it. But you have to recognize that inside that rock-looking structure are tissue, cells, and protein still there.
All right, so let's go back for a second, because if this horn or part of a dinosaur had been buried for millions and millions of years, you would not expect to still be able to see tissue. But are you saying that's what we're finding?
That is absolutely what we're finding. In fact, in a Nature Communication paper in 2015, they referred to it as common.
So is this unusual for those who have followed the traditional paradigm associated with when dinosaurs lived and when they died?
It would certainly be not at the least expected. In fact, Mary Schweitzer, who was the first one to really make this popularized, the first one to really get discoveries that were noticed by a wide range of scientists, she comments in interviews that she had her technicians repeat the study over and over and over again, simply because it's so difficult to understand how you could have this material still in a dinosaur fossil that is supposed to be 65, 70, 75, 80 million years of age. Because any competent biochemist knows that tissue, cells, proteins break down. They're not concrete, they don't just exist for eons of time, they break down. And in fact, they tend to break down fairly quickly depending upon the conditions. And certainly in Hell Creek, the conditions would be warm up, cool down, warm up, cool down. We found this horn, for example, just a mere 1 foot below the surface. Now, it was in very solid sandstone, so we had to chisel it out, but it was just below the surface. So there was no thermal protection by being deep in the ground. So it would have been very much subjected to fluctuations of hot and cold and hot and cold. And any biochemist can tell you that is the fastest way to destroy material. It's difficult enough to envision it surviving for 4,000 or 5,000 years, but 60 million years, 70 million years, see, that really becomes very difficult to make any kind of biochemical basis for how it could have survived.
Okay, well, let's talk now about what you do with these, okay? You've got these here. What would be your standard process of wanting to then examine?
Well, as you can see, these look like a rock. So what you have to do is you have to literally dissolve the rock away. So what we do is we soak the fossil material in a solution called EDTA. It's a very mild acid that will grab the calcium ions out of the fossil. And so you just literally dissolve the fossil. And what you'll have after you dissolve the fossil is the tissue will be remaining, because the EDTA won't dissolve the tissue.
So where are you now in this process? What would be your next step?
Well, some of these have just started, but the next step would be then we would take and we'd pour off the solution.
Okay.
Right now I'm not worried about what's in here. I will collect that and analyze it at some point later, but right now I'm just going to look at this.
So it's left there.
Right.
Okay.
So then I'll bring this over to what we call a dissection microscope. As you can see, what we see is this is, in essence, dissolved Triceratops horn magnified.
Well, Kevin, what did you find then when you were looking at the sample and you actually found some tissue?
Okay, here's what we found. Here we have a piece of the horn that has been decalcified, like what I just showed you over on the bench. Mark Armitage was our microscopist working on this, and Mark then took a piece of the decalcified horn and put it under a microscope. See the fibrous material there? That's part of the composition of the bone matrix itself. But what's really of interest is see the white material here on the surface swaying back and forth? That's actual dinosaur tissue on the surface. See, this is not a solid fossil. This has got tissue characteristic to it. See, notice how it flexes back. And that's, of course, very interesting, how you pull on it, flexes back, pull on it, flexes back. That's characteristic of tissue. That's what tissue would do. Now, Mark then was able to extract some very thin layers of elastic material away from the inner core of the horn. But he didn't have to decalcify the horn in order to do this. You can see it's stretchy, it's flexible. In fact, look, notice, see how it's stretching? It's stretching almost to twice its original size of what it was. See, this again is original dinosaur tissue that he's peeled directly from the fossil. There was no decalcification that he did first. See, this is how accessible this tissue was. He didn't have to remove any fossilized bone to get to this tissue. Okay, now, here is a light microscope picture of the tissue itself. You can see the texture of it. And in fact, see, notice the arrows, they're pointing to cells. These cells specifically are what we call osteocytes. Those are bone cells. They're involved in making bone, because even though we think of bone as a rock, bone is tissue. Bone is not a rock. In fact, in our bodies, bone is replaced about every 10 years to keep it all fresh and the new matrix laid down and such. So it's constantly being changed, and that's what those cells do. And if you look at them then at a closer magnification, what we see then, this is using scanning electron microscopy, you see the extreme detail of the cells. See how well that's preserved? I mean, that doesn't speak for something that has been degrading or something that has just been in a non-pristine condition for 65, 70 million years. We would not begin to expect to see such enormous and elaborate detail. I mean, these structures are incredibly small. You know, this is our 20 micron bar here. See how small these structures are, still intact. And yet, to see that kind of detail, then obviously the preservation process is surprising really to everybody. But I think as creationists, we have a lot less to explain than someone trying to suggest that this is, 65, 70 million years of age.
Well, let me ask you this first, because, I mean, this is an incredible picture. And I just want to make sure we're not looking at fossilized material here, fossilized tissue. That's been replaced by calcium.
These are the cells.
These are real cells.
Correct.
Well, Kevin, I understand you published all of this work.
Yes,
is that right?
This work has been published. We've actually made the cover of American Laboratory. We also published in a journal called ACTA Histochemica, and that's a more technical article. It goes through and explains then what we did, how we processed the horn, and then of course draws some conclusions from that. Particularly the conclusions are that even though it's a horn, which is different from a bone, it still had tissue. Even though it was wet when we pulled it out of the ground, it had what we call matrix, which is another word for mud. It still had tissue, so just a different specimen than what had been analyzed before. No one had done a horn before. Now we start getting closer, and what you're going to see then is you're going to see little pieces of tissue there, and that's what you're going to pick off and then put under the higher powered microscope. Okay, and then under the higher power microscope, see, now we look at the tissue itself, and there's the cells.
Oh, it's fascinating.
See, and then there's another. And like I say, those are very specific osteocytes. Now, those osteocytes have a very unique structure. Okay, now we're coming into some of the texture of the horn itself. And see, this would be a blood vessel. And if you look, see, this is dissolved away. This would be the interior of the horn. And you can see all the detail that'd be left in there after you take the calcium away. And then there, of course, is an osteocyte.
So that has to have shaken up the scientific community. What's been the response of all of this?
The initial response, when Dr. Schweitzer first published her work, which is what became very popularized in 2005, it generated a lot of response that previous papers had not generated. And there's kind of a question of exactly why it generated so much response. But in 2005, her paper was published in the journal Science, which is a very broad distributed journal, very highly respected journal. Color pictures, you can't minimize the impact of color pictures. So you had people that would be more biochemistry and biology backgrounds that maybe for the first time paid attention to this. So that generated, I think, some not only publicity for it, but certainly some controversy. And so initially some of the reaction was rejection. Oh, it's contamination. That's not really dinosaur. It's microscopic artifact. It's bacteria, because bacteria can look kind of strange sometimes. So you had a lot of proposals of what it could be. And to her credit, Dr. Schweitzer did more work, which is what science is. She did more analytical work, dug deeper. They began to find protein. You break open some of these cells. You look at the matrix these cells are attached to, and they're protein. Particularly one of the common proteins they found is called collagen. Now, collagen is the most common protein in any vertebrate. Vertebrate meaning those animals that have spinal columns. Collagen is the most dominant protein. It's a hardy protein. But there was no reason based on any kind of biochemistry known about collagen, any kind of biochemistry of how collagen degrades. There was no reason to think that collagen could naturally, easily survive for 65, 70 million years.
And all of that research, did it lead towards the conclusion that it's not bacteria, it's not something that
It very much
it?
did, right. That you can dismiss the bacteria idea, you can dismiss the contamination idea. It is real dinosaur tissue, real dinosaur cells, and real dinosaur protein.
Okay, so once that is understood,
yes,
then what happens? Now this is shaking it up, I guess.
That becomes part of the controversy, because clearly you're now faced with how could you explain the survival of this, the pristine survival of this, not only for so long, but in very unpristine conditions. There's nothing pristine about Hell Creek, Montana, for example. It's not permafrost. It's not like these were in a deep freeze for millions of years. Like we mentioned before, the temperature fluctuations, water. Water will degrade proteins. When we pulled the horn out of the ground, it had water underneath it just from the seepage of rainwater. That's why when we first dug the horn out, we thought, there's nothing going to be in there. And there was. So these are not dry, they're not sealed in some kind of, stainless steel vault. They're subjected to all kinds of conditions that would degrade this stuff. And so then the controversy has been, how do you explain it? And if you read some of the literature, there's almost like desperation of, would you guys please explain this? Because they recognized what the implications of this could be. Now, some people would claim, well, it means nothing because we know how old they are, and therefore it just means it survived somehow, big deal. But how do you know how old they are? Well, you used methods, supposed methods of dating. Well, this is a method of dating. The tissue itself can't be discounted as part of a method of dating. So why do you say that doesn't count, but this does count? Well, it's all the paradigm drives your conclusions. The paradigm is it has to be old. Therefore, methods that give us an old fossil are what we choose. Something that doesn't give us an old fossil, like tissue, we have to reject or explain away. And the big push at the moment is to explain it away, to come up with some explanation of how the tissue survived. There are several ideas out there. The most popular one at the moment is the one that Mary Schweitzer herself has proposed, where she proposed that in red blood cells you have hemoglobin, which of course is composed of iron, which is what then attracts and binds the oxygen, so that the hemoglobin in the red blood cell can transport the oxygen around in the body. Okay, what she's proposed is that upon the death of the animal, the red blood cells ruptured and they released the hemoglobin, which released the iron. In biological systems, iron can catalyze what's called a Fenton reaction. And this reaction, in essence, just causes proteins, for example, to cross-link. So it causes reactions of the proteins so that they actually become more resistant. So in this cross-link state, microbes don't degrade them as fast, enzymes don't degrade them as fast, they just simply don't compose as fast. And so she's proposed that that then explains how they could have lasted millions of years. We reject what she's at least proposed so far because we would say, first off, Fenton reactions are also going to leave signatures. They're going to leave signatures in how they're going to change the chemical state of certain amino acids. And in the protein analysis that's been done of like the collagen, for example, those amino acids in that protein don't have that altered chemical state that you would expect from a Fenton reaction. See, so we're not seeing the footprints that we would expect to see if these reactions were actually causing these massive changes to the proteins that were causing them to be preserved better. We would also say that the models themselves that have been studied take some of this into account. You know, the collagen models, they take into account some of the physical changes that are going to occur to collagen that you would say may make it more resistant to degradation. And yet the studies show that it still doesn't last tens of millions of years. So there is no physical chemical evidence that's going to support the idea that proteins, any protein, is going to be able to last tens of millions of years. It's just strictly an extrapolation. It must last because we know these are old. And there becomes your conundrum. Again, the paradigm driving the conclusion. We also would challenge that the study that Dr. Schweitzer did, she used ostrich blood vessels. And she soaked them in water, soaked them in solutions of iron from hemoglobin, soaked them in various solutions, and then monitored their degradation, how fast they degrade. And she reported that after 2 years, those that were exposed to the iron were, for the most part, undegraded. But first, 2 years at a steady temperature doesn't extrapolate to 65 million years at an unsteady temperature. Second, any technician can tell you that we take great pains in laboratories to preserve cells, to preserve protein, to preserve tissue. We freeze it. We deep freeze it. We freeze it, you know, minus 200 degrees in liquid nitrogen. You don't leave it out. You don't expose it to water. You don't expose it to all the things that, in all honesty, these fossils tended to be exposed to because everybody knows that accelerates degradation.
So in the normal sense, even someone who holds to a very recent creation, that would lead you to believe that this shouldn't be here either, right? Because even for several thousands of years, you wouldn't expect that.
It certainly would not be your first prediction. Even from a creationist position, we know full well that these fossils are exposed to ground level radiation. In fact, you could take almost any dinosaur fossil and put a Geiger counter against it and it'll light it up because they've absorbed radiation. So even for 4,000 years, we say, wow, that's still quite a challenge to think they'd absorb radiation and still. So how are you going to explain 65 million years of exposure to this radiation? And Dr. Schweitzer's iron preservation model doesn't account for that.
So as a microbiologist, when you look at this, the two major paradigms that we have before us, and even though this is surprising, there is a paradigm between these two that better fits the evidence than the other.
I think we understand enough about the process and enough about tissue itself to recognize that the more clear, parsimonious, if you will, the simplest explanation is simply that the fossils aren't as old as they're being claimed to be. And so that clearly, this is in violation of the dating process. It challenges the entire dating process. If the fossils of dinosaurs have been dated incorrectly, which I would say this is clear evidence they have, then it's very likely the fossils of any organism have been dated incorrectly, and therefore then the geologic ages themselves are incorrect. And we have to go back and recognize that they use evolution as their control, if you will. It's the filter. If I don't get a date that fits what evolution expects, then the date is rejected. It doesn't make a difference what the date is. It doesn't make a difference how you came about getting the date if it doesn't fit the filter of evolution, if it doesn't fit what we need. If you have something that's out of the Jurassic, but it's dated at 300 million years, that can't be right. Therefore, it's automatically tossed out. Why can't it be right? Because we have, in the evolutionary assumptions, determined that organisms that lived during the period of the Jurassic are this old. And so it sets then the interpretation for everything. When you have problems like the soft tissue, see, you either have to reject the entire dating process or you have to reject the soft tissue. You know, you really can't have both. They're trying to have both, but it clearly is one or the other. You know, either the fossils aren't as old as we think they are, or there's some mysterious, unknown, magical process that preserves them. Well, which is more scientific? What we know today, the tissue can't last that long, therefore the fossils can't be that old, regardless of what other dating methods you claim you've used.